Hi everyone. Today we will revise the topics, and the first topic that we are going to revise is DNA technology and human genome. So the first KPI 3.1.2, it talks about the role of DNA tools. So first of all, we need to know the DNA tools. So first DNA tool is restriction enzyme, and these restriction enzyme are very important. They cut, or you can say they cleave the DNA, and they produce either sticky ends or blunt ends. I will show you now. So imagine if this is DNA, okay, and the action of the restriction enzyme can be in two forms. So form number one, if it produces two ends with parallel two segments with the parallel ends, these are called as blunt ends. While on the other hand, if it is producing the DNA with hanging ends, like one strand is long, while you can see over here like the one strand is large while the second is small, and same over here, one strand is small while the second end is large. So such ends are called as sticky ends. So this is the basic difference between them, and the scientists, they prefer sticky ends. On the other hand, gel electrophoresis is a tool that is used to separate the DNA bands. So this is used for the separation of DNA based on two things. So it separates either based on charge of the DNA or size of the DNA fragment. So this is how it works. So another very important tool that we missed over here, this is called as the DNA ligase. So you need to keyword ligase, mean it glue or stick the ends, it glue or stick the DNA ends. So when you have the DNA ends and you want to glue them, you will use DNA ligase. So this is the basic description or the summary of the KPI. Now we will go to the question. Use the diagram below to answer questions 1 till 3. So we have here the most important thing that you need to notice is steps. So step 1, we have, sorry, okay, so we have step 1. Okay, it's not working on the image, it's okay, I will just go with the pen. So we have here step 1, then step 2, then step 3, and step 4, and these are the 4 steps that we will describe later in the image below. There is one more question where we have to describe them. Okay, so we need to know what is happening in step 1. So basically in step 1 you are isolating DNA. Okay, so this is the DNA from human, and this is the plasmid from the bacteria. So we isolate the DNA and the plasmid. So this is step number 1. And what is a plasmid? Basically, plasmid is a vector that we use to transfer the DNA from one organism to another organism. In step 2, so step 2 is what you see now, you cut them. So cutting is happening in step number 2. So this cutting is done by, of course, who? None than other restriction enzyme. So we cut it with the help of restriction enzymes. And the next one we need to know. So one more important thing over here, step number 2 is cutting, and we need to know, like, in this cutting what's happening. You can see over here in the cutting, you are going to cut the DNA as well as the plasmid. So you cut them, and in step number 3 you rejoin them. So this is rejoining, and this rejoining is done by this is done by the DNA ligase. And what we mentioned about the cutting is done by the restriction enzyme. Okay, now when you join them back, you can see that this has now two parts. So you can easily see over here that this is the part that is coming from the human, and this is the part, the lower white part is coming from bacteria. So that's why this is a plasmid basically, but now this is called as recombinant plasmid. And in the last step, that's called as insertion. So in the insertion, you reintroduce the plasmid back into the bacteria. Okay, so inserting, well, I can write it into in easy words. So inserting inserting back to bacteria. Now, when you inserting plasmid back to bacteria, when you insert plasmid back to bacteria, in this case, now this bacterial cell is called as a recombinant bacteria. So now this is not a normal bacteria. This is called as a recombinant bacteria. Moving to question number 1, which enzyme is used in step 2? So be very careful. Which step? So we already said in step number 2, restriction enzyme. So no need to worry more. If you know the diagram, you can easily answer it. DNA polymerase is an enzyme that's basically involved in the replication of DNA, and also we have studied that this is involved in the PCR reaction. DNA helicase, we didn't study this term, but this is known as the helix breaker, which breaks open the two strands of the DNA, and DNA ligase is the one that glue the DNA fragments. So when you have the DNA fragments, so you can glue them. Okay, which enzyme is used in step 3? We already mentioned up, if you remember, DNA ligase. We said that this is DNA ligase. So let's circle it out, DNA ligase. Which type of ends are produced in step 2? So you can see step 2 here, sorry. So you can see over here, this is a short side and this is a long side. Short side, long side. So this picture is basically like this. So if you see this picture, these are the two strands, they are like this. So okay, so one side is hanging while the other side is hanging. So of course now you know that the hanging ends are called as the sticky ends. So answer is sticky. We didn't study parallel ends, clear ends, we only studied blunt ends. When the ends look like this, they are 100% parallel, equal. Okay, so this is how the blunt ends look like, while the sticky ends look like this: one long, one short, and then one short, one long, like this. Interpret the data collected from the process of gel electrophoresis. Use the diagram below to show how DNA is separated to answer questions 4 till 7. So there is a diagram that's showing you the DNA isolation, or you can say extraction. And in this picture, we need to know there is this place is known as well, where you load your samples, load DNA samples. Okay, so you load samples over here, and this side, this side is a positive side of the gel, and the top is the negative side of the gel. And remember that the DNA is negatively charged, so of course it's going to move from the negative towards the positive end. So this is what we studied. So gel electrophoresis, it separates the DNA. That's what we said. So which of the following technique is used to separate the DNA? We said gel electrophoresis, so answer is C. Now, what are the factors which, on what basis it separates? So name two factors that are used to separate the DNA. So we already said size of DNA fragments. So you have size of the DNA fragments, and number 2, charge on DNA. So this is how it separates them. Which of the following is correct about the two fragments labeled as X and Y? Okay, so first of all, X. So remember that the larger fragments are toward the top and smaller fragments are toward the bottom. So X is larger than Y, and all of them are negatively charged. Each one of these lines is called as a band of DNA. Okay, this is the thing. So X is negatively charged, yes, and it is bigger than Y, that's yes. The remaining you will find smaller is wrong. X is positively charged is wrong. Which two children are most likely to be the biological siblings and are children of one of the father and the mother shown in the diagram? Okay, how you figure it out? You need to make sure that you match the bands. Okay, so how you match the bands? You just go in matching. Okay, so let me just do this. So this is the mother father band. So father band is matching with child number 1, okay, only, while the second band of the father is matching with child number 2. Okay, so 1 and 2. Now for the mother, the band is matching with 1, 2, but it's not match, it's very close, but it's not matching with 4. Be very careful, okay? And then the second band of the mother is not matching with anyone, so it's quite obvious now that it is matching only with 1 and 2. Okay, so 1 and 2 is the correct answer. Moving to the next page, sequence diagrams and models how recombinant DNA is made and manipulated. We discussed already how the recombinant DNA is made. Recombinant DNA is made in 4 steps. If you remember from the picture on the first diagram, step number 1 is isolating the DNA. Step number 2 is cutting the DNA. Step number 3 is rejoining, and step number 4 is inserting back into the plasmid. So we go back, okay, and which of the following is the correct sequence of the steps in the process? Okay, so we need to look for cutting. If you don't have cutting, you will have cleave. Okay, cutting, then so first was isolation. Okay, remember isolation is not given over here. No worries. Okay, if it is not given, it mean missing. You can check the remaining in the sequence. Cleavage, joining, introducing. Yes, that makes sense. Joining is not before cleavage, introduction is not before joining, and cleavage is after cleavage. It should be joining, not this one. So all other are wrong. The same diagram. Okay, so we know that what are the steps. So first step is, we can write them down here. We can write them down here. So step number one is animal DNA and bacterial plasmids. And what I told you, isolated plasmids are isolated. So in step number 1, this is what's happening. Step number 2, both of these are cut with, remember to say, with same restriction enzyme. If you don't use restriction enzyme, you will lose the half mark. In step number 3, the DNA fragments, the DNA fragments of animal and plasmid, what is happening over here? Join, right? Joined by DNA ligase. If you don't say DNA ligase, you lose another half mark. And the last step we said introduction, right? Now this is recombinant plasmid, so you will say that recombinant plasmid is introduced back into bacterial cell. Okay, bacterial cell. So this is all what's happening in the 4. Define diagram, and the diagram shows an example of the recombinant DNA technology. Define recombinant DNA. Recombinant. So a DNA that results from joining two segments of DNA, but make sure that which segments of DNA from two different organisms. Remember, it will be only recombinant when you have different organisms. Okay, that's it. We move to the next. Detailed using the diagram of the PCR. PCR stands for polymerase chain reaction. Is written right down here. okay, and this is one of the processes that's very important to produce millions of the copies of the DNA, and that's the purpose which we have to write here. Okay, so remember, they're asking you purpose, okay, to produce millions of copies of original original DNA. What is the name of the component binding with the DNA strand in step 2? Okay, this is step 1, step 2, step 3. So step 2, this small segment, you can see, find this small segment over right away here. See, so it's quite easy to answer it. That's the DNA primer. So what's the name? DNA primer. That's it. There is no worries about that. What's happening? So in step number 1, what's happening? You are breaking open the two strands of the DNA. Step number 2, primer is joining. Step number 3, DNA polymerase is extending. And every single process of the polymerase chain reaction, when one cycle completes, each cycle each cycle results in two copies. Each cycle results in two copies. Okay, complete the table below to explain the process of. So I told you step number 1, name the step. So step number 1 is denaturation. Step number 2, annealing. Step number 3 is extension. Step number 1, what's description that's taking place over here? That the two strands of the DNA are separated. So two strands of DNA are separated by heating. Also, you can say the hydrogen bonds are broken. Hydrogen bonds are broken. And this process takes place between any temperature 90 to 96 degrees Celsius. In annealing, we already noticed that the primer binds with each strand. So when we say the primer bind with each strand, then you can easily say that this process takes place between 50 degrees Celsius to 65 degrees Celsius, any temperature you can say. And the last step, extension, the DNA polymerase. But remember, there is a very specific polymerase, that is Taq polymerase, okay? That is only involved in step number 3. Taq polymerase adds new nucleotides to DNA strands, okay, so to complete the two copies, and this process takes place at 72 degrees Celsius. Better stick to the 72, okay, that will be good for you. Next one, define the term genomics. It's straightforward question. Genomics is the study of organism's genome. Okay, so that's the answer. While we will go through the other options as well. The computer applications that stimulate genetic experiments. So this is bioinformatics. And the remaining two we have not studied, so just skip them. Identify the goals of the Human Genome Project. So what are the goals? Of course, the goals were to facilitate the development of the new therapies. Yes. Sequencing the entire genome. Yes. 3 billion nucleotides. To correct the mutated genes that cause human disease. No, this is gene therapy. It's not the goal of that. So this is not. And to identify all human genes, yes, and we want to choose which one of the following is not. So it means that the answer is C. So the answer is C, because this is not the goal. Specify the applications and benefits of the gene therapy. Just now we said what is gene therapy, but what's the application? Let's see it. Cystic fibrosis is an inherited disorder that causes several damages to the lungs, digestive system, and other organs of the body. Which of the following tool would be better option for treating individuals who are affected with cystic fibrosis? And that's what exactly say it over here. to correct the mutated genes that cause human diseases. So this is the definition of the gene therapy. So that's the answer, gene therapy. Identify the aim of the gene therapy and one way in which it can be done. Okay, so we just now noticed that what's the aim. So the aim is gene therapy aims to correct the mutated gene. Okay, so this is the aim. And how it do. Okay, so second part, remember there are two parts. Okay, it can be done by inserting a normal gene into cells to replace the non-functional or mutated gene, you can say non-functional gene.